Thank you for taking the time to learn about what we do at a botanical garden. When my neighbors ask what I do, and I tell them that I work at the New York Botanical Garden, they usually ask about their houseplant, which is usually not in great condition. So I tell them what I do, that I don't work in horticulture, but that I'm a scientist and work on the evolutionary genomics of plants. And they usually say, "What do you do?" But then I also tell them that I don't have a green thumb and that my husband does not let me take care of the plants in our own apartment. And then the conversation is usually done. But I wanted to share with you a little bit about the breadth and depth of research we do at the New York Botanical Garden and how the models developed during the Herbarium 2020 challenges are crucial to our current research. My basic take-home message is that botanical gardens play an integral role in stopping extinction and coming up with solutions to combating the climate crisis and food insecurity. But first, I wanted to thank all of the participants of the Herbarium 2020 Challenge hosted on Kaggle. I hope you really enjoyed the data set, and I hope you get excited about all of our curated collections and what we could do together. A huge debt of gratitude to Serge Belongi, Christine Kaiser-Chen, and Kiak Kwan-Tan from Visipedia and Google for shepherding the Herbarium 2019 and Herbarium 2020 competitions on the Kaggle platform for the fine-grained visual classification workshop. I also want to acknowledge funding and support from the New York Botanical Garden and the National Science Foundation. But I particularly want to give a huge thanks to all of my colleagues and to all of the explorers before them that collected the plants for this competition and for being the best stewards of the plants of the world. The botanical research that I'm going to tell you about should come as no surprise. There is a long and deep history of discoveries made in plants that apply to all life. But what surprises many people is how little we know about the world around us. There is a link below to a blog about some of the discoveries made in plants and how they are fundamental to all life. For example, on the left is Robert Hooke and how he coined the term cell after studying cork. And on the right is Gregor Mendel, the father of genetics, who discovered the patterns of inheritance by just studying pea plants. There are more discoveries using plants as research organisms, and these discoveries include epigenetic inheritance, transposons, the mutation theory, and more. The New York Botanical Garden is a museum, and we have collections. We have a lot of collections. We have living collections found out throughout the grounds and in our glass houses. We have herbarium collections, anatomical collections, pickle collections, tissue collections, slide collections, and DNA collections. And we see ourselves as the stewards of the plant kingdom. One of the main areas of research at the New York Botanical Garden are discovery and exploration. It is still surprising to me and most people that we are still discovering new species of plants. New York Botanical Garden's main area of exploration are the Americas and Southeast Asia. Shown on the left is my colleague Doug Daly collecting in the Amazon, and on the right is my colleague Fabian Michelangeli collecting in Peru. NYBG scientists alone discover about, on average, 50 new species of plants every year. My colleague Andrew Henderson discovers new species of palms each year. These are not small, inconspicuous plants, but trees. And I think this illustrates how little we know about the world around us. So how do you determine if a species is new? Well, it helps to be an expert in the group that the plant might belong to, because you need to know what has been described already. But you can start by looking at key features such as reproductive structures, how those are arranged on the plant, vegetative structures like leaves and how those are arranged. We call this broadly morphology, or the study of plant form. Another area of research is determining the relationships of the plants to each other. And for this we use plant form and the different characters of the plant, as well as DNA sequences. And we can also understand the distribution, once we know where those plants occur, the distribution of those plants, and try to better understand how these plants have evolved. My own area of research is plant evolution and development. I use all of the different types of collections for my research. I study patterns of plant diversity at all levels, from the gene to the organism to groups of organism to global patterns of diversity. I investigate how leaves, fruits, and flowers are made, from a single cell to a multicellular organ. This is called the development of the structure. But I also investigate which genes build those structures. So which genes are necessary to build a leaf, which genes are necessary to build a fruit, which genes are necessary to build a flower. And then I look at those genes and how differences in those genes can help to explain all the diverse forms that we find in leaves, flowers, and fruits. So on the left is just an illustration of all the different forms of leaves that we can find in ferns. And on the right, in purple, we're looking at where one particular gene is turned on within the plant. And these types of analyses can help us to better understand how changes in genes can give us changes in form. And all of this may seem very, sort of esoteric and out of bounds, but think about, what did you eat today? What did you have for breakfast? What did you have for lunch? I had Earl Grey tea and Nutella on toast. Those are all leaves and fruits. So these are really relevant to your everyday life, and who knows what we might find through these types of investigations. There are other questions that I'm interested in about plant evolution and development, and one of our projects is called Living Fossils. This is a really large collaborative project with the New York Plant Genomics Consortium, which includes the New York Botanical Garden, New York University, American Museum of Natural History, Cold Spring Harbor Labs, Johns Hopkins, and other institutions. And one of the questions that we're looking at is living fossils. And the reason we're looking at this is that because plants can't move. They've been around for a long time, and they thrive under diverse environmental conditions. So what solutions have plants come up with to survive over hundreds of millions of years of evolution, and how can that help us survive? Living fossils is a term coined by Darwin, and I'm showing here 2 living fossil species, Metasequoia on the left and ginkgo on the right. I just want to give an example of what we're studying in this project. You can find out more on the webpage livingfossils.org. But if we're going to look and try to understand how these species survive, we look at Ginkgo biloba. And ginkgo is a living fossil. And there is a fossil that was found that is dated to nearly 300 million years ago. And that fossil of ginkgo looks virtually the same as the extant tree you see growing on the sidewalks of cities. To put that sort of time frame, 300 million years, into better context, the first dinosaur fossil dates to around 240 million years ago, and dinosaurs went extinct 65 million years ago. So for nearly 300 million years of changing temperatures, changing CO2 levels, continents breaking apart and crashing together, mountain ranges rising, Ginkgo biloba persisted and remained virtually unchanged on the outside. But something had to have been happening, so we suspect that there's something in its genome that makes it particularly resilient. So the question is, what does an adaptable genome look like? And we are comparing ginkgo to its sister taxa. So this comparative approach becomes really strong, and we're comparing it to cycads. So even though cycad fossils could be dated to nearly 300 million years ago, all of those species went extinct, and all of our extant cycad species recently speciated 5 million years ago. So it looks like cycads had a completely different evolutionary strategy. So we will compare the genomes of ginkgo to cycads, but we're not just comparing this pair, we're comparing different pairs of living fossils to more recently speciated sister taxa across the gymnosperms. And the gymnosperms are particularly important because they're the main component of boreal forests, and these are the first forests we will lose to the climate crisis. And the last really cool thing I wanted to tell you about this project is that plant genomes tend to be really large. The human genome is about 3 billion base pairs. Ginkgo is nearly 12 billion base pairs, so nearly 4 times the size of the human genome. And pine is nearly 20 billion base pairs, so more than 6 times the size of the human genome. So we're coming up with tools and different analyses to sequence and better understand these genomes, and the tools that we develop for this particular project to understand what adaptable genomes look like, what recently speciated genomes look like, but the tools that we develop for these analyses will be applicable to genomes across all living organisms. So on the flip side to living fossils is extinction. And although there are plant species that persist for a long time and we can learn from these, there is an imminent crisis. And I'm not talking about the climate crisis directly. We already knew that we were currently in the 6th major extinction event. What is new this week is that the pace of extinction is happening much faster than we thought. So this article, published in the Proceedings of the National Academy of Sciences this week, took a look at a small portion of vertebrate species, and this story was picked up by the New York Times, CNN, The Guardian, and other news outlets. But that article just looked at a small fraction of vertebrate species. Just last year, a couple of papers were published that showed that plant species were going extinct much faster than animal species. And another manuscript used herbarium specimens to show that we actually lost more plant species than we thought. And so if you add the plant data to the vertebrate data, then you realize how dire the situation is. And also, if you consider the charismatic fauna that are going extinct, then you need to consider their habitat and their food sources, which are plants. There will be no pandas if there is no bamboo, and there will be no hummingbirds if there are no red flowers. So no species exists in isolation, but in a finely balanced ecosystem that includes plants, animals, fungi, and billions of microorganisms. And there will be a domino effect with extinctions, and the extinction rate will quicken. So I just wanted to show you, this is a phylogenetic tree illustrating the relationships of organisms. There is a single origin for all of these organisms. We have 1.7 million known plants, animals, and fungi, and there are predicted 40,000 vertebrates. That is all your mammals, reptiles, amphibian, fish, and birds, 40,000 altogether. There are 400,000 known plant species, 400,000. Those plants create habitats for all of those animals, including us. We are a single species on this tree right here. We are a single species within those 40,000 vertebrates, within this tree of 1.7 million known plants, animals, fungi, and one. And it is because of that one terminal, us, that we face an extinction crisis, a climate crisis, and food insecurity. But if we are to solve these crises, we need to look more holistically at the planet. And we can. We can look at species occurrences of plants around the planet. We know where plants have occurred. This is a map of Captain Cook's voyage, and we can know the time and year that the plants that were collected were making flowers and fruits, and we can look at the occurrence of plants and their phrenology and how it may have changed over time. We have occurrence data for all of our collections. So during Captain Cook's voyage, during these 3 years, Joseph Banks made collections of plants from around the world. And some of these collections are now stored in our own collections at the New York Botanical Garden. In fact, at the New York Botanical Garden, our herbarium has more than 7.8 million specimens. And as I showed you before, it's a lengthy process of documenting the flora of the world, what it is, who is it related to, and where can it be found. But on the other side, there is a dearth of taxonomic expertise across plants. There are 400,000 known plant species, but there are 80,000 estimated to still be discovered. 80,000 new species of food, shelter, and medicine. And to go from the field to species identification, it is estimated to take 35 years from field collection to pressing to identification. We have a room at the New York Botanical Garden with 30,000 unmounted, undetermined specimens. During Herbarium 2019 and Herbarium 2020, were developed so that we could more quickly curate these species. We need models with anomaly detection so we know if a specimen is new, so that we can get that specimen to the correct taxonomic expert. Herbaria are considered a major frontier for species discovery, with an estimated 50,000 of undetermined specimens already present in herbarium collections as new species. As I mentioned before, herbarium specimens are very rich in knowledge. So in addition to determining what species are out there, there's much information on the specimens. Plants are collected when they are reproductive. They have dates of collections and locations. And we can also extract DNA from these dried collections. So each specimen is a wealth of information. But not all the information is easily extracted. On the right, Lycopodium reflexum was collected on the 21st of March, 1997. It's collected in Puerto Rico, and we have exact GPS coordinates at 18 degrees, 16.5 minutes north and 65 degrees, 46 minutes west at an elevation of 1,000 meters. The habitat it was found in was terrestrial and on steep banks along the roadside. And it's all typed out and easily read. But if you look at the specimen in the middle, it's handwritten, Lycopodium reflexum. It was collected on the 19th of March, 1920. It was collected in Jamaica on a road from Silver Hill Gap at 900 meters to Hardware Gap at 1,200 meters, but its habitat was steep, brushy bank. So trying to figure out the exact GPS coordinates of that location is a little bit more difficult, and even reading the name can be difficult. On the left, the species name is handwritten again, Lycopodium limnifolium. It was collected between 16 and 17 of July 1905, collected in Haiti on Camp Number 1 Corral at 1875 feet. And its habitat was on a tree and it was pendant. So even though we have a lot of metadata we can extract from more current specimens, we still have some new tools we need to develop to really better understand the location data of some of these specimens. So we need new tools for species discovery. We need tools to better understand global patterns of plant diversity, not just understanding the diversity of one small group of plants, but understanding that global pattern of all plants. With our specimen records, we should be able to start reconstructing where plants occurred, how the timing of flowering and fruiting has shifted over time, where to focus our efforts in plant exploration. Imagine if we can overlay these patterns of plant diversity with climate data and soil data. I think that all of this is possible, and it's absolutely necessary to combat extinction and to help us better confront the climate crisis and food insecurity. Thank you very much for your participation in Herbarium 2020 and being part of the solution towards saving the plants of the world.